Welcome to Part 3 of Adenovirus in North American Cervids. Dr. Leslie Willis Woods received her DVM and PhD in comparative pathology from UC Davis and is a diplomat of the American College of Veterinary Pathologists. She is a professor of clinical anatomic pathology at the California Animal Health and Food Safety Laboratory at the School of Veterinary Medicine at UC Davis. She's been at the Veterinary Diagnostic Laboratory in California for 36 years and was at the Wyoming State Veterinary Diagnostic Laboratory for a year in 2006 and 7. She has 15 publications on the deer adenovirus, including the first paper documenting the disease.
I see lots of familiar names, so I just wanted to say hi, everybody. I'm going to give a quick update on the adenovirus hemorrhagic disease in deer in California. And throughout their presentation, I'm going to ask questions because just like Jennifer said, I have more questions than answers on this disease, even after studying it all these years. And I hope maybe that the discussion afterwards, everybody can talk about their experience and maybe solve some of these. So the next slide just demonstrates the pulmonary edema and the hemorrhagic enteropathy that was demonstrated by Jenna and Jennifer. But so I call it an enteropathy because there isn't an itis. I think it's a DIC. And so it's mostly hemorrhage in the intestinal tract. DIC, just like the pathogenesis of the orbiviruses. And next slide. So what we didn't see was the localized infection in these animals that were presented, which is necrotizing almost abscesses from the mouth to the forestomachs. And the next slide demonstrates in a percentage of the cases, it does not go systemic. They essentially, the infection is localized, and based on one experimental study, it didn't even go systemic first. It was localized infection. And so you do see the vasculitis here and then a thrombosis, and then you get the tissue dying that's supplied by the vessel, and then you get lots of secondary bacterial infection. So you can isolate several different kinds of aerobic and anaerobic bacteria from these lesions, including Fusobacterium necrophorum, which we know can be primary, but in this case, it is secondary. And as I said, this form of the disease does not go systemic in a percentage of cases. So as previously noted, checking the lung or the spleen for adenovirus by PCR is worthless. Even going to the lesion and checking for adenovirus by PCR or immunohistochemistry is fruitless in a large number of these cases because these animals live longer and the virus is long gone or destroyed by the secondary bacterial infection by the time you get these animals. During the big 93-94 outbreak that we had in California, we did suspect that adenovirus was the cause of the localized infection. I think we had about a 50% localized, 50% systemic, but we weren't able to prove it until we got the 11th deer in which we were able to demonstrate the adenovirus. We also had a 50-50 percentage in outbreak in a captive flock here on campus, a captive herd here on campus. And in that particular case, we had 50% localized, 50% systemic. And even in those animals that were right here on campus, we weren't able to demonstrate the adenovirus because it just takes longer to die. But we do know the adenovirus is responsible for the localized because we have reproduced it. The last point I want to make is you can get the combinations of the systemic and the localized. But in those animals, you'll have the pulmonary edema and you'll have ulceration through the upper alimentary tract, but it'll be more acute ulceration. So next slide. And we called this virus otocoileus adenovirus type 1, but Myrna Miller proposed cervid adenovirus type A, and it appears that this is the more correct name based on her isolation from elk and your cases in elk. We do have Thule elk and Roosevelt elk in California, and definitely the mule deer. overlap the range of the elk, but we have not had a single case in elk in California. And then the next slide, we did next three, yeah. Next one, yeah. So we did, back up a little bit. We did reproduce this fulfilling Koch's postulates. The important points that I wanted to make that I learned from the experimental studies was that it is transmitted by contact, nose to nose. It doesn't need a vector, which is what you'd expect with the adenovirus. You can go to the next slide. I haven't gone out and collected insects, and then the next, and then the next. I haven't gone out and collected insects, and tested by PCR for adenovirus, but adenoviruses are not typically transmitted this way. Could be transmitted mechanically, I suppose. So the virus lives in the environment for a long time, depending on conditions, and it's very contagious. And I also found that it's very rapidly shed once an animal is infected. Okay, so the next slide. Let's see, next slide. Let's see, backup one. I think I'm frozen.
Okay.
Sorry about that. Okay, so questions for discussion now. So the outbreak in California in 93, 94 was big, and it back up. I'm sorry, it's yeah. You know, over thousands of deer died in over 17 counties in California, and I think like it's been similar along in Oregon, maybe Washington. So maybe Colin and Kristen can talk about that. But in other states, it doesn't seem to be as they don't seem to have as big an outbreak. And why is that? We really don't know, but there is a bit of evidence from Myrna Miller's study. She did sequence 10 isolates from deer in Wyoming, Washington, also the elk and in Colorado from you, Karen. And with that study, she also sequenced the isolate from the 93-94 outbreak in California. She did find evidence of two distinct clades, and there was a difference between elk and moose. It's very, very close, but there was a difference in a variable region, non-coding region. And in the California 93-94 outbreak isolate, she found that there was a 21 base pair deletion in that same region, which is interesting. I mean, it's N of 1, so we can't make any definitive call on that, but it is interesting information. And then the next outbreak, on the next slide, sorry. Might be frozen again.
But we're having some network connectivity issues for sure.
Oh, yeah, there is a big storm in California too. So when we went back through the archives at the California Department of Fish and Wildlife, we found that there was another huge mortality in 1986, 87, and then also in 83. And then we tested those tissues, and they had, we detected the adenovirus in these tissues also. So another question for discussion is, where does this virus go between the outbreaks? You know, these are the big questions. You know, so possibilities are it's ubiquitous and most deer are infected but are asymptomatic. And mortality events occur when there are stressors such as starvation from drought or overpopulation. Or you can have a few asymptomatic carriers. They intermittently shed and antibody wanes in most of the deer in herds, and they become more susceptible. Or there could be a wildlife reservoir. So next slide. So we touched on the question of, can other species become infected and shed? is there a domestic reservoir? I mean, this virus is very closely related to bovine adenovirus 7 and ovine adenovirus 7. So we did studies in colostrum-fed and colostrum-deprived calves and lambs, and they do not become infected and they do not shed. And then we checked some wildlife reservoirs. We tested tissues from mice collected from counties during deer mortality outbreaks in those counties. And we tested the tissue, and none of the tissues in these rodents were positive by PCR. And then we also inoculated ground squirrels, and they do not become infected and they do not shed virus. And the reason we checked the mice and the squirrels was because the biologist mentioned that there seemed to be a population explosion of these animals during big outbreaks, so we had to check. So a retrospective study, next slide. A retrospective study demonstrated that this is an important disease in California. There is a difference of opinion among biologists on whether disease is an important factor that can impact populations. But one of our biologists that was convinced that it is showed me some data that a data set that he was working on for the hair loss syndrome. And it showed a sustained impact on his deer populations following big adenovirus mortality years. And they didn't seem to recover, at least in his herds. And then the next slide, last of all, I want to show you some seroprevalence of AHD in California. So serum was collected from 500 deer in 10 counties from 1983 to 2002. And we sent it to Howard Limcool, who did the serum virus neutralization test. It's difficult to look at the whole picture because sampling was intermittent in some herds and not done in others. And it depended on funding that year and biologists' time and interest in deer. So this is one of the reasons it's been probably difficult to publish this information. But next slide shows that we were able to test 471 serum samples from free-ranging deer. And then the next, just click once. Okay, so, and as it turns out, we had only 30 of the 471 that were positive for antibody to the deer adenovirus. And years and counties with seropositive animals correlated with mortalities that were diagnosed in these counties due to the deer adenovirus. And so the last figure right here, the number of samples that we tested from this county and the number of positives, and I highlighted in yellow the years that we also made a diagnosis of the deer adenovirus hemorrhagic mortality. And then the very next slide, what was interesting is there are three subspecies of mule deer in California that seem to have a seroprevalence, and we got no reports of mortality in these deer. You know, so I'm thinking maybe these subspecies are less susceptible because they were, the California mule deer did overlap with their deer herds. Okay, so the Round Valley, next slide, the Round Valley herd in Mono County was the most complete set of serum samples that we did have because they were doing a 20-year study and they collect serum every year. And these were Rocky Mountain mule deer. So I think that we can make a good story out of these samples. If you look at 1986, which remember there was an 86-87 outbreak, we got 2 positives. And then we had 1 positive on the backside of the 86-87 outbreak. And then we had 4 positives in 94. And all the rest of them were negative. You know, so in my mind, that means that seroprevalence is low based on at least serum virus neutralization test. So either the antibody doesn't last long or maybe the most infected deer don't survive. So I'd like to hear the epidemiologist comments on this data. So it doesn't appear that asymptomatic infection is common in most susceptible mule deer species. It's not ubiquitous, you know, say like BVD in bovine. So next slide. And then the next one click thing. So then we tested lymph nodes to answer this question. Are most deer latently infected with the virus, and then stress triggers shedding and clinical disease? So adenovirus infections in humans are usually self-limiting, and they frequently establish a latent persistent infection of the tonsils and lymph nodes. And seroprevalence in human adenovirus type 5 infection is about 60-70% in Europe and in the US, and it's 90% in Africa. So if the deer adenovirus is hiding in the tonsils, retropharyngeal lymph nodes, similar to human adenovirus, I would have expected seroprevalence to be high, but we found out it's low. So we wanted to test the lymph nodes. These were lymph node pools that actually were collected for chronic wasting disease, and then we put them in the -80 and then ran them for adenovirus by PCR. And what we found was, next click, is only 4 of over 1,000 tested were positive. And these were from counties during years that were currently experiencing the adenovirus hemorrhagic disease mortality. So I think the serological evidence and the PCR surveys might indicate very few deer are persistently infected. And these are likely the source of virus when stress triggers shedding, and that most deer don't have neutralizing antibodies to this virus, assuming it is protective. And therefore we have our outbreaks. And so next slide is just a number of questions and just a couple of clicks to get the whole slide to show up. And next, and next, and next. And then one more. And then we'll be done. I think we will. Anyway, we still don't know what triggers the really big die-offs. Is it an environmental issue? Brant Shoemaker looked at weather patterns and our big outbreaks, and he didn't find any correlation. The genes, we have that N of 1 from Myrna that says maybe there's a difference in the sequence of the isolates that caused the huge outbreaks. And then we do know that at least during the 93-94 outbreak, there was a management issue. We had one of the rehab centers that was funneling lots of fawns through. They had an outbreak, and then they released to multiple counties. And then where does the virus go between the outbreaks? Do we have rare deer that are persistently infected and they're the source? Do we have a wildlife reservoir? We really haven't ruled that out, just looking at 2 species. But I'd love to hear everybody's comments and opinions about some of this in the following discussion. So that's all I have.
Please tune in for Part 4, where members of the Wildlife Health Working Group of the Western Association of Fish and Wildlife Agencies will share their experiences with adenovirus hemorrhagic disease in wild cervids.